In this video, we'll discuss about monohybrid, dihybrid and trihybrid crosses and how to find out the phenotype and genotype ratios of these crosses. After we are done with all three crosses in the orthodox method, that is by plotting Punnett square and actually counting the number, I'll give you a short formula by which also we can reach to those ratios. So let us talk about the crosses first. The monohybrid cross is what we are talking of. Monohybrid cross. And as we have already discussed in the terms that monohybrid means when we are taking only one trait or one character into account. So, and we will start with the typical Mendelian way, that is with pure line. So our say parent generation is homozygous tall crossed with pure recessive, that is homozygous tall with homozygous short, or pure dominant with pure recessive. The gametes produced by these plants or organisms would be each one with the dominant allele, and here each type of gamete would have the recessive allele. And in F1, if we plot the Punnett square, the ratio that we get after placing these gametes here is going to be 4:0. Let us fill this Punnett square. The dominant allele from one parent and recessive allele from the other. And the same way we are going to get 4 offsprings. So here in F1, the ratio is going to be 4:0, whether we talk of phenotype ratio or genotype. So our phenotype ratio is 4 tall and none short. And genotype ratio is going to be 4:0 again. 4 are heterozygous tall. Now on selfing, when we get the F2 ratio, we don't have to worry about F1 because according to the law of dominance, in all F1s, all offsprings are going to be showing the dominant trait. But if we go to F2 on selfing, that means if we cross the members of F1, we will get the F2. That means heterozygous crossed with heterozygous. The gametes produced would be one with dominant allele, one with recessive, and with this also same. And if we plot the Punnett square here, this is the ratio which we are looking for. So gamete of one parent on one side, other parent on the other side. So here it is going to be both the dominant, that is homozygous. These two will be heterozygous and this one is going to be homozygous recessive. So if we talk of phenotype ratio, that means how they look. This will look tall, this will also look tall, and this will also look tall. So here the ratio is going to be 3 tall and 1 short. So 3 are tall is to 1 short. And genotype ratio means genetically how their genetic makeup is, that is genotype ratio. This one is homozygous dominant. These two are heterozygous. So 1 is to 2, and this and the corner one is homozygous recessive. So genotype ratio is going to be 1:2:1, whereas phenotype, that means on the basis of their appearance, the ratio is going to be 3:1. This is what we get in a typical Mendelian cross when we are talking of monohybrid cross. Now let us take a dihybrid cross and reach to the F2 ratios, that is phenotype and genotype F2 ratios. I'm going to just leave it here and we will write down what type of ratios are in case of monohybrid cross, so that when we write that small formula, it will help us. In F1, let us write the ratios like this. F1 phenotype ratio. In F1 all were tall, so it was, and genotype ratio genetically also. In F2, the phenotype ratio means the way they looked, the offsprings, was 3:1. Phenotype ratio was 3:1 and genotype ratio was 1:2:1. This was what we got in case of monohybrid. Now let us talk about a dihybrid cross. In a dihybrid cross, we would be taking two characters into account. And again, we will start with the typical Mendelian way. So parent generation is going to be pure for both. So if we say pure tall, that is dominant, with purple colored flower, is crossed with pure recessive, that is for short, and white flowers. In F1, well, let us see what type of offsprings or ratio we get. The gametes produced by this plant will be with T and P, and all four gametes are going to be of same type. Here also with recessive allele, that is for short, and recessive for color, that is white, and all four are going to be of the same type. And if we plot the Punnett square here to find out the ratio in F1, let us place TP, same type of gametes, showing dominant allele here, all four showing recessive. So the offsprings will have one dominant, one recessive for height, one dominant, one recessive for the color. And all 16 offsprings are going to have the same. Here also capital T and small t, capital P and small p. So when we fill up this, we will find that all 16 offsprings are tall and purple. So if we write phenotype ratio, how are they going to look? They all are going to be tall with purple colored flower. So it is going to be 16:0. All 16 are tall and purple. Now, if we talk about the genotype ratio. What is the genotype of this individual? It is having one dominant allele for height and one recessive. That means it is heterozygous tall and heterozygous purple. So here also ratio is going to be 16:0, but they are heterozygous tall and purple. So phenotype, genotype ratios are same in case of F1 generation. Now we want to see F2 generation ratio. On selfing of these, what ratio do we get in F2? So let us write down the F2 ratios, or rather find out. So heterozygous crossed with heterozygous. What type of gametes are produced in this plant or organism? Capital T here, that is dominant with dominant P. The second one would be capital T with small p. Third one, small t with capital P and both small. So four types of gametes are possible by both these plants. So now let us write these gametes here and find out the genotype, phenotype. Capital T, capital P, capital T, lowercase p, small t, big P, small t and small p. Same here, T and P, capital and small, small capital P, and these are all small. Now here we will fill up the Punnett square. This is going to be having both dominant for T as well as P. Here is going to be T and T, capital and small, capital T, small t, capital P, capital P, capital T, small, capital and small. Here T's are capital, P and P. Both T's are capitals, P's are small, one T capital, one small, one P capital, one P small, and here T is capital, other is small, and the P's are all small. Capital T, small t, both the P's are dominant. Here capital T small, big P and small p. Small t, P and P. Here both the T's are small, one P is capital and other small. Here T and T, P and P. This one T and T, small p, small t, capital P and small, and here all four are going to be small. So now let us find out the phenotype first, the phenotype ratio. We are talking of how they look, phenotype ratio. So let us see how many are tall and purple. For tall and purple, there should be at least one dominant T and at least one dominant P. So this will be tall and purple. This also has capital T and P, so tall and purple. This also tall and purple. This one also. Here also. This has T, but the P's are small. So this one tall and purple, not this. Tall and purple. This one also tall and purple, not this one, because here T is not dominant. Same here. This one also is going to be tall and purple. No capital P, no T, and this one. Let us count how many. 1, 2, 3, 4, 5, 6, 7, 8, and 9. So there are 9 which are tall and purple. Now let us count how many show only tall as a dominant trait and the other trait is recessive. That means tall and white. This one will be tall because it has T but no capital P, so this will be tall and white. This one again same, tall and white. No, this one, it has capital T and small, so tall and white. Okay, so there are 3 which are going to be tall and white. Now let us count the other dominant, that is short and purple fur, which is the dominant trait. So this one, this is not having capital T, so it will be short, but it has capital P, so this will be purple. So this will be short and purple. Same here, this will also be short because of recessive T's and purple. This one short because of small T's or recessive T's, and P is dominant, so short and purple. Not this one. So 3 are going to be short and purple. And this last one, it has all genes as in their recessive form. So this one is going to be short and white. So our phenotype ratio is 9:3:3:1. This is in a typical Mendelian cross, the phenotype ratio is 9:3:3:1. And we have written the dominant on this side, maximum dominant, and this end has mostly the recessive ones. Now, the 9 here, they show both dominant characters. 3 show one dominant for height and one recessive. The other 3 show the recessive for the first one and dominant for the second. And the last one shows all recessive traits. So this is our phenotype ratio. Now how do we reach to genotype? Genotype means heterozygous and homozygous. So this is homozygous for tall and homozygous for purple. This is homozygous for tall and heterozygous for purple. So if we keep counting like this, it is going to take lots and lots of time. So I will give you a shortcut or a formula by which we can reach to the conclusion of genotype ratio. So we'll write the genotype a little later. Before that, we want to understand how to plot the crosses in a normal orthodox method, so that in case if we forget the formula, then also we should be able to reach to that answer. And that is why we are taking the orthodox method first, so that we understand the concept, and then at the end I'll give you the formula. So I'm leaving this genotype because it is going to take too much of time. I'll give you a shortcut so that we can reach to the formula or a ratio, genotype ratio, quickly. So let us write down phenotype ratio in a dihybrid cross here. So this was our monohybrid cross. Now let us write down dihybrid cross. In F1 we got all 16 phenotype ratio 16:0 and genotype ratio was also 16:0. All 16 were tall and purple and all 16 were heterozygous tall and heterozygous purple. In F2 we have found out phenotype ratio which is 9:3:3:1. Genotype ratio is what we will write down after some time. Now let us come to trihybrid cross. Trihybrid means we are taking 3 traits or characteristics into account. Say we are talking of pure tall with purple flower and say the position also. Axial position of the flower crossed with pure recessive for all 3. That means short with white flower and with terminal position of flower. So we have taken 3 traits. We have taken height, we have taken color of the flower and we have also taken the position of the flower. This becomes our parent generation. In F1, what ratio do we expect? Here, the number of gametes are going to be 8. So 8 gametes of one parent on one side of the Punnett square and 8 gametes on the other side. So we will get 64 offsprings in this cross. Let us first talk about the gametes. One out of these 2 T's, capital, because they are both capital T's. One out of these 2 P's, both are dominant, so capital and A. So all 8 gametes are going to be of same type. So this is 8 type, 8 number. Here also recessive, recessive and recessive. And all are going to be same type. So there are 8 gametes of the same type. Now, if we plot Punnett square, and again, in F1 we don't have to plot the complete Punnett square. If we write lowercase t, p and a here and all gametes here, they are going to be all same on this side and all same on this side. So offsprings will be one dominant allele and one recessive, one dominant for color, one recessive, one dominant for position and one recessive. That means all 64 are going to be of the same type. So if we want to know the phenotype ratio in F1, it is going to be 64:0. And all 64 offsprings are going to be tall, purple and with axial position of the flower. Genotype ratio, all are going to be heterozygous. So 64:0 and all are heterozygous, tall, purple and with axial position of the flower. Now, if we have to find out the genotype of all these 64 offsprings in F2 or phenotype in F2, again, that is going to be a time-consuming process. But we will find out what type of gametes would be produced in F2 or offsprings so that calculation becomes easy. Now we are crossing members of F1 to get the F2 ratio. So members heterozygous for all 3, capital T, small t, capital small, capital A and small. Now, how many gametes are produced? Number is 8 and types are going to be, again here 8 types. How many types? 8. Number 8. In F1, type only 1, but the number of gamete was 8. Why 8 types? 1 out of this, 1 out of this and 1. So we have to write down all possible combinations. We'll start with all dominant. Capital T, P and A. 3 dominant. Now we will come to 2 dominant. T, P dominant, A small, T, A dominant P small, P and A dominant T small. Here we have taken two dominant. Now we will take one dominant, T dominant, P and A small, P dominant, T and A small, A dominant, T and P small. And one more option is there, all three recessive, T, P and A. So 1, 2, 3, 4, 5, 6, 7 and 8 types of gamete. Many a times they ask questions on the basis of the genotype. How many types of gametes are possible? I'll give you a short example here. Suppose if we talk of T, T, P and A. This is a variation from this. Here it is homozygous for all but hetero for these two. So how many types of gametes are possible? Capital T with P and A. Then capital T with small p and capital A. Capital T with capital P and small a. And then capital T with small p and small a. How many types are possible here? It is not 8 types, but the number is going to be only one, that is 8 gametes are produced. So we have to remember that in a trihybrid cross, number of gametes are 8 always, but the types can vary depending upon the genotype of the parent. Now 8 on this side and the same 8 on this side, and we will get 64 offsprings in F2 generation. We can find out the ratio by plotting the Punnett square also, but as we are preparing for exam, and in exam we don't have time to plot this is going to take about 20-25 minutes to fill up this square. So there is a short formula by which we can reach to this number. Let us cross check using the formula these two crosses which we have already made. Now to find out phenotype ratio, the formula is 3:1 raised to n, where n is the number of traits or characters which we have taken into account. In case of a monohybrid cross, our general formula will be 3:1 and n here in a monohybrid is 1. So 3 multiplied by 1 or 1 multiplied by 1. It is going to be 3:1. So are we getting the same ratio here? So in case of monohybrid, our phenotype ratio by the orthodox method came as, or we found out it to be 3:1. And if we calculate by using the formula, then also we are getting the same. And we are talking of phenotype. Then in dihybrid, the formula remains the same, 3:1, but this n is now 2 because we have taken a dihybrid cross. This basically means 3:1 into 3:1. How do we reach here? 3, we'll have to use this 3, multiply 3 with 3 and 3 with 1, and then 1 with 3 and 1 with 1. So 3 into 3 is 9, 3 into 1 is 3. We are done with 3, multiplication of 3 with these two numbers. Now we'll multiply 1 with these two numbers. So 1 into 3 is 3 and 1 into 1 is 1. Are we getting the same ratio here as we got by plotting the Punnett square? So this is phenotype ratios that we have talked of. Let us talk of a trihybrid phenotype ratio now. In a trihybrid phenotype ratio, we can have a shortcut. Shortcut means same 3:1 and n, and because it is a trihybrid cross, this n is going to be 3. That means 3:1 into 3:1 into 3:1. We just now calculated this. This came to 9 is to 3 is to 3 is to 1. Now let us multiply this with this number. So now 3, we'll multiply all these numbers with 3 and then all these numbers with 1. So now let us start with 9 into 3, 27. 3 into 3, 9. 3 into 3, 9. And 3 into 1, that is 3. Now let us do the same thing with 1. 1 into 9, 9. 1 into 3, 3. 1 into this 3, 3. And 1 into 1 is 1. We can reshuffle this ratio to 27 is to 9 is to 9. We can bring this 9 here. So 3 times 9 and then 3 times 3, 1, 2, 3 and 1. This is our phenotype ratio in case of a trihybrid cross. So phenotype ratio will be 27, then 3 times 9, then 3 times 3 and 1. So by using the formula, we are able to reach to this number. But we also need to remember the normal orthodox method in case if we forget this formula. That's why we discussed the normal method first and then I'm giving you this formula. Now let us calculate the genotype ratio using another formula. So for genotype ratio, the formula is 1 is to 2 is to 1 raised to the power 9. N is again the number of traits which we have taken into account. So, in case of a monohybrid cross, it's going to be 1 is to 2 is to 1 raised to 1. That means the ratio is going to be 1 is to 2 is to 1. Is this the ratio that we got here? Same. So, now we know that orthodox method and the formula also gives us the same. Let us talk of a dihybrid cross. In dihybrid cross, same thing. 1 is to 2 is to 1 raised to 2 because now the traits which we have taken into account are 2. This means this multiplied with 1 is to 2 is to 1. So ratio is going to be 1 multiplied with 3 numbers, 2 multiplied with these 3 and 1 multiplied with all these. So let us write down 1 into 1, 1. 1 into 2, 2. 1 into 1, 1. Now doing the same thing with 2. 2 into 1, 2. 2 into 2, 4. 2 into 1, 2. With 1 again, it's going to be 1 is to 2 is to 1. This is the genotype ratio in a dihybrid cross. And that is why we left this blank here. So it is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Now let us do it with a trihybrid cross. So this is your dihybrid ratio. 1, 2, 1, then 2, 4, 2, and then 1, 2, 1. In case of a trihybrid, our formula is going to be 1 is to 2 is to 1 raised to 3 because now we are taking 3 characters into account. This is what we get in case of a dihybrid cross. That means we'll have to multiply this with 1 is to 2 is to 1. This is going to be a little long ratio, but let us quickly write this. We'll multiply 1 with all these numbers. So the numbers are going to be same. 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Now the same numbers multiplied by 2. So 2 into 1, that is 2. Then 2 into 2 is 4. Then again 2 into 1 is 2. Then 2 into 2 is 4. Then 2 into 4 is 8. Then again 2 into 2 is 4. And I'm continuing here. then 2 into 1 is 2. 2 into 4 is, 2 into 2 is 4, and then 2 into 1 is 2. And then we have to multiply all these numbers with 1. So here it is going to remain the same. 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. This is going to be our trihybrid genotype ratio. Now that is why, as I said, we can plot the Punnett square, but to find out such kind of a ratio manually, there can be errors. So in case of a trihybrid cross, we can depend on these general formulae by which we can reach to these ratios. So in case of a monohybrid dihybrid cross, phenotype, and we are talking of F2 ratios, because in F1 all offsprings produced are going to be showing dominant traits, and they are all going to be heterozygous for whatever traits they are showing. Phenotype is 3:1, genotype is 1:2:1 in F2. Here also in F2, the ratio that we have to remember is 9:3:3:1. This is phenotype. Genotype ratio is this, which we have calculated by using this formula. And in trihybrid cross, the ratios are calculated again by using these formulae. So this is just a shortcut, and these are standard, because whenever we talk of mono, dihybrid and trihybrid crosses, we always start with the Mendelian principle. And if we start with Mendelian, the end result is going to be same. So that is why this formulae, they help whenever we are appearing for an exam.